Hello students, welcome to EPG Pathshala. I am Dr. Zeenat from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard. Today we are going to talk on the module Nasal Drug Delivery System, and we would be looking forward to understanding the per se site of action and the other related basics. This is from the paper NDDS 1. So my dear students, let us see what we are going to learn in this module. The first thing, as of now, we would be going in for the introduction. We will have a brief understanding of various drugs which are available in the market, which are preferably given through the nasal cavity. Then after this, we will spend enough time on understanding the physiology of the nasal cavity. Later on, we would also spend time on understanding the factors which will affect the drug availability or nasal bioavailability per se. Towards the end of this module, we would try to understand the various advantages and disadvantages of the nasal drug delivery. My dear students, let us try to appreciate a simple fact. If you just name a few drugs of the categories like mucolytics, antibiotics, decongestants, there will be a large number of drugs which are preferably given through a nasal cavity. So if we just try to understand one important point, that there can be two varieties of drugs which can be delivered through a nasal cavity. We can have something which probably is for local effect and something probably which is for the purely systemic effect. We have got various types of administration devices. We can have nasal sprays or we can have nasal drops which are probably there only for the localized effect. Then later on, if we just try to go into the details of it, we can probably find a large number of drugs which are preferably given to the site of this nasal cavity so that the effect is much pronounced. We should appreciate as pharmaceutical students that we can deliver both the hydrophilic as well as the lipophilic drugs, and which can be easily absorbed through the nasal mucosa. A classical example of a drug which can be administered intranasally and for a treatment of the disease like migraine is sumatriptan. The other classes of drugs which are successfully administered by this route include steroids. We can exemplify them by the names like estradiol, progesterone, testosterone. Then a very important category of the drugs which probably are referred to as peptides are something which are preferentially delivered through the nasal cavity. The question arises, why is it so? We also understand as pharmaceutical students that peptides are not suitable to be delivered through the oral route. Reason being, there would be a large amount of stability issues. So, the drugs like buserelin, desmopressin, calcitonin, and insulin are preferably delivered through the nasal cavity. Then we have another category of drugs exemplified by nifedipine, nitroglycerin, propanol and hydrolazine, which are together as antihypertensives, can also be delivered through the nasal cavity. My dear students, before we get into the nuances of the nasal drug delivery systems, we need to understand the physiology of the nasal cavity. The nasal cavity extends from the nostrils to the nasopharynx, that is the throat, and is divided in two major regions. Laterally, by the nasal septum. Each region of the nasal cavity opens through the nostrils, which then again extends posterior to the nasopharynx. Both the symmetrical halves of the nasal cavity consist of four areas, that is, the nasal vestibule, atrium, respiratory region, and olfactory region, which are distinguished mainly according to the histological as well as the anatomical properties. The total surface area of the nasal cavity in humans is found to be about 150 centimeters square, and the total volume is estimated as about 15 ml. The two regions of nasal cavities are subdivided into different regions such as nasal vestibule, superior turbinate, inferior turbinate, middle turbinate, olfactory region, sphenoidal sinus, frontal sinus, and cibriform plate of the ethmoid bone. The nasal vestibule presents the smallest cross-sectional area in the respiratory tract, which extends from the entrance of the nostrils and are guarded by vibrissae to anterior ends of the inferior turbinates. The vestibule lining changes from skin at the entrance to squamous epithelium and then to ciliated columnar secretory epithelium at the turbinates. The walls of the nasal septum are folded to form turbinates and metases. Metases are primarily the air spaces. The narrow space of the metases, that is between 0.5 to 1 mm, helps in maintaining proper contact between the airstream and the epithelium nasal passage lining. The lining of the olfactory region of the nose consists of non-ciliated neuroepithelium and is located towards the roof of the nasal cavity. The remaining passage of the nasal cavity is lined with pseudostratified columnar secretory epithelium consisting of basal cells, goblet cells, and columnar cells, which may be ciliated or unciliated. Microvilli are present on the columnar cells, which increase the surface area required for drug absorption. The nasal mucosa presents a highly vascularized area. The superficial and deep layers of arterioles supply the lamina propria. There are numerous sinuses or venous lakes present between the venules and capillaries, which are linked to erectile tissue, particularly in the middle and inferior turbinates, which enable the airways to widen or narrow. This automatically controlled vasculature of the nasal tissue in combination with its rich supply of secretory cells is of importance in the modification of the inspired air. The olfactory region of the nose is located towards the roof of the nasal cavity, which is lined with non-ciliated neuroepithelium. This region captures approximately 20% of the air flowing through the nasal cavity. A large number of bipolar neurons are present in this region, which reacts with the inspired air, resulting in initiation of impulses in the olfactory nerves. A very important aspect of nasal cavity is mucociliary clearance. This primarily contributes to the defense mechanisms of the body by entrapping potential hazardous substances such as dust and microbes within the viscoelastic mucous blanket lining the nasal passages. The secretory product, which is mucus, is then propelled by cilia, followed by pronounced beating in a coordinated manner within the periciliary fluid towards the nasopharynx, where the mucus and any entrapped particulates is either swallowed or expectorated. Periciliary fluid is something to be understood as a watery fluid maintained by transepithelial ion transport, which provides a favorable environment within which the cilia are able to beat, provide a reservoir of fluid for the required humidification of inspired air. When the periciliary fluid becomes too deep, it is assumed that the ciliary tips would be unable to reach the mucous layer, and hence the clearance would therefore be compromised. When the periciliary fluid becomes very shallow, the cilia becomes entangled in the mucous gel and mucus transport would again be compromised. Let us now discuss in detail about the composition of mucus its role, and other aspects. It is well understood that the nasal epithelium is covered by a mucus gel. The goblet cells and the serous cells glands present in the nasal cavity secretes approximately 1.5 to 2 liters of mucus. The mucus plays a number of important physiological roles which can be summed up as: number 1, it entraps various substances that enter the nasal cavity and participates in the removal of particulates via mucociliary clearance. This process actually protects the underlying mucosa. Another important role which the mucus plays is that it has got a very good property or capacity to hold water, which permits and helps in better humidification of the inspired air and thus aids the heat transfer. Mucus is primarily composed of water, which is around 90 to 95%, salts, which constitute 1 to 2%, lipids, about 30% of the non-aqueous secretion, and mucins are 0.5 to 5%, which actually gives the gel-like properties. The composition of the nasal secretions includes glycoconjugates, which primarily include the mucus glycoproteins, glycosaminoglycans, proteins like albumin, IgG, secretory IgA, lactoferrin, lysozyme, and non-secretory IgA. It also has a substantial quantity of proteases, which primarily are neutral endopeptidase, aminopeptidase, peroxidase, and carboxypeptidases. It also has lipids, primarily exemplified like phospholipids, triglycerides, free fatty acids, and cholesterol. Besides this, it also has the nucleic acids like DNA and RNA. The protein content of nasal secretions comprises of the following composition. It has the secretory IgA, which helps to prevent the attachment of microorganisms to the mucosa, lysozyme, which attacks the cell walls of microbes and hence acts optimally at the acidic pH. The pH of the nasal secretion is generally ranging between 5.5 to 6.5, but it becomes more alkaline during bouts of the common cold. Lactoferrin is also present, which sequesters any kind of free iron present in the secretions, which might otherwise be used by iron requiring microorganisms. Besides this, it has got the various other enzymes. The respiratory mucins are long and linear high molecular weight glycoproteins, which are polydisperse in the mass. They are like flexible threads which vary in length ranging from 0.5 to 10 micrometers. Mucins are composed of subunits which are referred to as monomers, each about 500 nanometers in length. The monomers are joined from one end to the other end via the disulfide bonds. Each monomer comprises of a protein backbone core with alternating oligosaccharide-rich regions which are approximately 100 nanometers in length, and the naked regions of folded proteins are further stabilized by the disulfide bonds. The protein backbone is cloaked by the oligosaccharides, which are rich in the amino acids such as serine, threonine, and proline. The naked protein regions are additionally characterized by high cysteine content, which provides the required sulfur molecules for disulfide bonding. Mucins comprises of high carbohydrate content approximately 80% by mass, which are present in the form of short O-linked oligosaccharide chains. Serine and threonine provides adequate sites of O-glycan attachment through the sugar and acetyl galactosamine linkage and the oligosaccharide chains, which may also contain N-acetylglucosamine, galactose, fucose, sialic acids, and sulfate molecules. Mucins also contain minute amounts of N-linked oligosaccharides, which are found to be important in the correct folding of the naked regions of the core protein. Along with it, 7 mucin genes MUC1, 2, 3, 4, 5AC, 5B, and 7 are found to be expressed in the respiratory epithelium. Mucus possesses both elastic as well as viscous properties simultaneously and therefore is assumed to be as a viscoelastic gel. The viscoelastic properties of the mucus arise from the non-covalent interactions, which are also referred to as the entanglements, between the predominant anionic mucin molecules, although weak hydrogen bonding and ligand-like attractions between protein regions of adjacent molecules may also play a role. Gel properties of the secreted mucus in the nasal cavity are affected by various factors such as the size of the mucin, polydispersity of mucin, and the type of the mucin present. Required amount of specific rheological properties must be possessed by the mucus for providing efficient clearance from the airway. Pronounced alteration in the specific rheological property compromising the mucus clearance can lead to various types of airway diseases or nasal infections. Various agents are capable of altering the rheological properties of mucus. The agents can be categorized in two groups. The first groups are known as mucolytic agents, which include N-acetylcysteine, carbocysteine, mercaptoethanosulfonate, and bile salts. The second group of agents are known as mucospecifics, which includes DNA, bacterial alginate, tetracycline, Congo red, and chlorhexidine. The level of mucin hydration can also have a significant effect on the properties of gel. Viscoelasticity of mucus is increased in pathological conditions such as asthma, chronic bronchitis, chronic obstructive pulmonary diseases, and acute respiratory distress syndrome. Increased viscoelasticity of the mucus secretion and its poor clearance occurs due to dehydration of mucus, which are mainly observed for the patients with cystic fibrosis. Therefore, the behavior of the nasal mucosa will directly govern whenever there is the requirement of drug delivery through this route. The pathological conditions would definitely have an impact on the final behavior of the drug delivery and ultimately its bioavailability when delivered through the nasal cavity. The next important things which we should understand is related to the nasal bioavailability. What are the most important things which we should understand here? Out of the many, the one which is of paramount importance are the physiological factors. There are a large list of physiological factors which will actually affect the nasal drug bioavailability. The nasal cavity, by virtue of its nature, is usually supplied by a large quantity of secretions. There would be a particular volume which is being present there continuously. So when we are talking about the first factor which will affect the nasal drug bioavailability, it is probably the secretions from the nasal cavity. We have a nasal cavity, and this nasal lining is usually having a large amount of cilia, or the fiber like, or the hair which we talk about. So we have another important factor which is physiologically present there and which will probably affect the drug bioavailability, and this is referred to as the mucociliary clearance, and which also includes the ciliary beat frequency. Then whenever we are talking of the drugs, it is not, of course it is understood that the situation of that individual is diseased, or the person is suffering from some pathological disease. So the pathological conditions of the nasal cavity will also become an important factor in the process of the drug being available. Next, we come to an important factor, that is your nasal membrane permeability. As for when we had been talking about the other mucosal surface, the nasal membrane itself is a mucosa. It is naturally present inside the nasal cavity, so there will be some amount of permeability which is naturally present there. So what we call this is the nasal membrane permeability, and again it would be affecting the nasal drug bioavailability. The nasal cavity is probably highly vascularized cavity if we talk it in terms of the physiology and the anatomy. So blood supply would be present there, it would be present in larger quantities with much more velocity. So blood supply also becomes an important factor which will affect the nasal drug bioavailability. Then we will have another factors like the enzymatic activity, immunological clearance, and the mucus clearance, which are supposedly the factors which will definitely affect the nasal drug bioavailability. My dear students, we just had talked about the various physiological factors. Let us now take each of these factors individually and try to understand that how they are present and what can be the effect on the drug delivery. The first factor is the secretions of the nasal cavity. Nasal secretions are produced by anterior serous and seromucous glands. Permeation of the drug through the nasal mucosa is affected by the various factors. The first factor which will affect the drug delivery permeation is the viscosity of the nasal secretion. The viscous surface layer affects the mucociliary clearance of the administered drug if the mucus layer is either thick or thin. If it is thick, it will be impeded, and if it is thin, it might be a little faster. Solubility of the drug in the nasal secretions is the next important factor. For permeation and absorption of the drug, solubilization of the drug is the primary step. The administered drug needs to have appropriate physicochemical properties for the required dissolutions in the nasal secretions. The next factor which primarily will affect is the diurnal variation. The nasal clearance of the drug is affected by the circadian rhythm of the body. Permeation of the drug is altered at night due to the variation in nasal secretions, and hence clearance rate of the drugs are usually very much reduced. The next factor is the pH of the nasal cavity. The pH of the nasal cavity primarily varies from 5.5 to 6.5 in adults and 5 to 7 in infants. Ideally, the pH of the formulation should be between 4.5 to 6.5 for better absorption in the nasal cavity, and should also have good buffering capacity to produce a better effect. The next factor which we would discuss is the mucociliary clearance as well as the ciliary beat frequency. The mucociliary clearance acts as a normal defense mechanism in the nasal cavity, and it clears a nasally administered drug in nearly 21 minutes by draining it into the nasopharynx. Reduced mucociliary clearance increases the contact time between the drug and the mucous membrane, leading to enhanced drug permeation, while increased mucociliary clearance leads to decreased drug permeation. The site of deposition affects the mucociliary clearance rate of a drug molecule. Let us try to understand that what is the actual effect. The mucociliary clearance is high for the particles which are deposited on the ciliated regions. The clearance is somewhat slow for those particles deposited on the non-ciliated regions. Particles that are deposited on the nasopharyngeal regions are swallowed immediately and are hence not available for nasal absorption. This small discussion actually focuses on that how important the mucociliary clearance is when it comes to the delivery of the drug through the nasal mucosa. Now let us talk about the pathological conditions and how they will hamper or affect the drug delivery through the nasal mucosa. Permeability of nasally administered drug is adversely affected in several pathological conditions such as mucociliary dysfunctioning, hyposecretion or hypersecretions, irritation of the nasal mucosa in disease conditions such as the common cold, rhinitis, and nasal polyposis. The common cold consists of two phases. What is that consisting of mucus hypersecretion, and the second phase usually is nasal congestion. Less than 10% of intranasally administered drug remains in the nasal cavity after 25 minutes, whereas approximately all drugs remain up to 90 minutes during the nasal congestion phase. Thus, it is evident that the behavior of the drug delivery would drastically change in presence of pathological conditions as simple as that of the common cold. This probably will lead to unpredictable absorption profile of the administered drugs. Such kind of problem may be resolved by addition of a vasoconstrictor in the formulation, which may relieve such kind of symptoms and exhibit pronounced absorption of drug. We would now discuss the next important parameter, and that is the nasal membrane permeability. Permeation as well as absorption of the drug is highly affected by the membrane permeability. Water-soluble and high molecular weighted drugs such as proteins and peptides exhibit low membrane permeability, hence are absorbed through endocytic transport mechanisms in lesser amounts. We now move on to the discussion of the next parameter, and that is the blood supply. The nasal mucosa, as we all know, is highly vascularized. This unique property plays a good role in the process of drug absorption in the nasal tract. High vascularization leads to higher drug absorption, leading to rapid onset of action of the nasally administered drug. This actually eventually becomes one of the major advantages of the nasal drug delivery systems. The next important parameter which is going to affect our drug delivery through the nasal membrane is the enzymatic activity present there. Various enzymes such as neutral endopeptidase, aminopeptidase, peroxidase, and carboxypeptidases are present in the nasal epithelium. These enzymes are responsible for low bioavailabilities of some nasally administered peptides due to enzymatic degradation in the nasal cavity. When peptides are administered intranasally, they are exposed to both intra and extracellular enzymes. When the peptide is absorbed by the paracellular route, the action of intracellular enzymes remains insignificant. Exopeptidases, which includes aminopeptidases, carboxypeptidases, and dipeptidyl peptidases, cleave at N and C termini, whereas endopeptidases like encaphalinase and cathepsin B cleave at an internal peptide bond. Action of endopeptidases is significant for larger peptides, whereas smaller peptides are resistant to their action. Although it is known that the enzymatic activity in the nasal mucosa is lower than that of the gastrointestinal tract, this route remains as an alternative route to the oral delivery of enzymatically labile drugs such as therapeutic peptides and proteins. The activity of the enzyme cytochrome P450 is higher than that of the liver and is responsible for metabolizing the inhaled pollutants into reactive metabolites, which are responsible for inducing nasal tumors. We move on to the next important parameter, that is the immunological clearance. It is well-known fact that various antibodies are secreted in the nasal cavity in high concentrations, which have the ability to neutralize the antigens present in the nasal mucosa. Antibodies have been reported to be detected in the nasal secretions in response to intranasal administration of insulin. Insulin is susceptible to degradation in the nasal cavity due to the action of nasal enzymes. The patient eventually may suffer from allergic diseases such as hay fever. The pharmaceutical excipients included in the nasal formulation, which causes some kind of nasal inflammation, might enhance such kind of reactions, and hence it is important that the nasal formulation excipients are selected with great caution. We now come to an important factor which probably will play a monumental role in the drug delivery through the nasal cavity, and that is the mucus barrier. Mucus affects the drug delivery by either posing a physical barrier to drug diffusion or by binding to the drugs. The absorption of intranasally administered human growth hormone is enhanced by the use of mucolytics. Increased binding of drugs to mucin may result in limited diffusion. So the presence of the mucus, the barrier properties of the mucus are to be considered while the nasal drug delivery formulation is under development and designing. We have just in the previous few minutes have discussed in detail the nasal physiology, the various factors which will be affecting the drug bioavailability. One thing is very important to note here is that despite so many barriers, it definitely has got very clear-cut advantages. The first advantage, which probably we will all appreciate, is that this is a completely and totally non-invasive method, and this is also additionally having an advantage where the onset of action is much more rapid. The second type of advantage which probably is offered by this type of drug delivery system is it has got a rapid absorption of drug due to the presence of highly vascularized nasal mucosa. The third and a very important factor as far as drug delivery is concerned is this is the route of choice when we are having the drugs which are susceptible to hepatic first pass metabolism, and when we are giving such drugs through the nasal cavity, by default the hepatic first metabolism is avoided. Then we can also very conclusively say that another major advantage would be that the drug bioavailability is definitely enhanced. The drugs which cannot be given by oral route or the parenteral route, of course, will rely upon this particular route as delivery system. And these can be very well administered by the nasal route. Not to be probably overlooking it, we can have a large list of advantages, but definitely there are certain limitations also. So let us try to sum up the various limitations of nasal drug delivery system. When we talk of pharmaceutical drug discovery, we will have a huge variety of drugs which will have smaller molecular weight or bigger molecular weight, which is an inherent property of the drug. We cannot control that. So one limitations which this route is giving is that the drug which are having higher molecular weight, maybe around let us say 1 kilodalton, it is not suitable for the delivery through the nasal cavity. So this becomes not necessarily a disadvantage, but we can say that this is a limitation. Then because of the presence of the nasal mucosa, it can result in the moment, for example, if you're putting a drop or you're putting a spray, then there is a large amount of possibility that it might result in nasal irritation. So that actually becomes a huge limitation. Then most of the time when I'm talking of the nasal drug delivery, there is a situation that a person is suffering from common cold, is suffering from flu. So one of the major symptoms in such conditions is that the person will have a congested nasal cavity. So in case of congested nasal cavity or in pathological conditions which are similar to this, the absorption of the administered drug will be greatly affected. So again, we can say that this is a limitation of this route. Then huge interspecies variability in drug absorption is observed in this route. We can probably then have this thing in mind that it would not be much different from one human being to another human being, but it has been very clearly noted that it usually does also have an inter-subject variability. My dear students, up till now we have talked in detail about the various parameters which will affect the nasal drug delivery. We also were able to understand the various advantages and limitations of the nasal drug delivery systems. Let us now try to understand a very important aspect, and that is the various characteristics of the ideal drug candidate which would be suitable for nasal delivery. It is not to be overemphasized that all drugs would not be suitable for such a system. Therefore, there are certain characteristics of the drug which will make it as an ideal drug candidate for delivery through the nasal mucosa. These characteristics include: number one, the high aqueous solubility of the drug is desired, which will provide the needed dose, which could be between 25 to 150 ml volume of the formulations per nostril. Then, the less irritation in the nasal cavity is an important parameter to be considered. That means the drug in no way should be able to cause any irritation. The drug should also give a good nasal absorption profile. There should be a very cogent and clearly understood clinical rationale for the designing and choosing of such a drug for nasal dosage forms. What is also desirable is the less metabolism of the drug in the nasal tract and avoidance of the formation of toxic metabolites in the nasal epithelium. It should have very suitable stability characteristics, and of course it should not have any offensive odor or aroma which is associated with it. So, students, we have come towards the end of this module, and let us now try to summarize what we have learned in this module. The very first point which is to be remembered and which is a take home message is that intranasal drug delivery can serve as an alternate route for drugs that are having some problem if they are to be delivered intravenously, or some problem if they are to be delivered through the oral cavity. For example, in case of the oral cavity, the drugs are absolutely not suitable to be delivered because of their physical properties, chemical properties, or their compromising on the therapeutic properties. So we have a choice that I have a drug which is having a high hepatic metabolism, so I by choice deliver this drug through the nasal cavity. The second important take home message is that this route is appreciated because it is rapid, it is non invasive, and in case I want my drug to reach to the CNS system, so it also has got that capacity, if formulated properly, to bypass the BBB or the blood brain barrier. We must appreciate, while we have understood the physiology in this module, that it is highly vascularized in nature. The nasal cavity has got a huge unique physiology. It serves an excellent site for absorption of both hydrophilic as well as lipophilic drug, and it can be very suitably used not only for loco-regional delivery, but it can be very well exploited for the systemic circulation delivery as well. The nasal drug delivery is affected by a number of factors. This is one important take home message, that until and unless we are able to appreciate these factors, we will not be in a position to design the most suited and the most favorable kind of drug delivery system. So let us just very quickly summarize these factors which are going to affect the system. The first is the secretions of the nasal cavity, naturally occurring, nothing much can be done. We have to take this into account and then make our system. We have the second, again because of the physiology and the anatomy, the mucociliary clearance, then the ciliary beat frequency, the pathological conditions, the nasal membrane permeability, the blood supply, enzymatic activity, immunological clearance, and the mucus clearance. What we can in conclusion say, that the nasal physiology has to be understood. These factors which will affect the drug bioavailability are to be considered if we have to deliver and design a very successful nasal drug delivery system.